Right now, your body is a contested resource. You share your environment with a massive array of microscopic pathogens, viruses, bacteria, fungi, and parasites, all looking for a warm, nutrient-rich place to multiply. For most of human history, treating a severe infection required desperate measures. Doctors would administer heavy metals and harsh chemical poisons like arsenic. The goal was to kill the invading disease slightly before the poison killed the patient. Modern pharmacology faces a much tighter constraint. The challenge is designing a lethal chemical that systematically dismantles a microscopic invader while leaving the human cells right next to it completely unharmed. In the early 20th century, a scientist named Paul Ehrlich conceptualized a theoretical ideal for this kind of medicine. He called it the magic bullet. Ehrlich's idea evolved into the governing principle of antimicrobial medicine: selective toxicity. To build a magic bullet, pharmacologists have to find and exploit specific biochemical structures that exist inside the pathogen but are entirely absent from human biology. By cataloging the exact molecular differences between humans and microbes, scientists have developed 3 primary strategies to sabotage an infection. The first strategy targets the outer perimeter. Human cells are enclosed by flexible, fluid lipid membranes. Many bacteria, however, rely on a highly pressurized, rigid outer cell wall to maintain their shape and survive. Pharmacologists exploit this reliance using beta-lactam antibiotics, a class that includes penicillin. These drugs specifically inhibit the synthesis of peptidoglycan, the structural material bacteria use to weave their outer armor together. As bacteria attempt to grow, their structural crosslinks weaken. Without an intact wall to hold back immense internal pressure, the cells rupture. We apply this same logic to fungi, which use beta-glucan. Echinocandins dissolve that material, collapsing the fungal fortress while completely ignoring human tissue. If an invader relies on building materials that human biology doesn't use, we can engineer specific molecular compounds to shatter them. When we can't breach the perimeter, the second strategy is to attack the interior assembly lines. To spread an infection, microbes must rapidly replicate their DNA and synthesize new proteins. The enzymes that pathogens use to unwind and copy their genetic material perform the exact same function as human enzymes, but their physical shapes are slightly different. This diagram shows how fluoroquinolones, like ciprofloxacin, exploit that difference. You can see the quinolone drug wedged directly into the bacterial DNA gyrase, the enzyme unwinding the strands. This physically locks the double helix in place, stopping replication cold. We use a similar jamming technique against viruses. Viruses are difficult to target because they hide inside our own cells, hijacking human replication machinery to survive. To fight back, we use drugs like nucleoside reverse transcriptase inhibitors, or NRTIs, critical in treating HIV. These drugs act as counterfeit genetic building blocks. As the virus builds a DNA chain, it blindly grabs this counterfeit block. Lacking the correct attachment point, the entire viral assembly line jams permanently. By feeding an infection incompatible structural parts, we force the invaders to sabotage their own replication from the inside. The third strategy abandons physical structures and targets a pathogen's metabolic supply lines. Consider folate, a nutrient essential for producing DNA. Humans absorb folate passively from the food we eat. Many bacteria, however, lack the ability to absorb it. They must synthesize their own folate from scratch using specific chemical precursors. We exploit this by administering sulfonamides. These drugs closely mimic the bacteria's natural precursors. When the microbe attempts to use the drug to build folate, the chemical pathway fails, and the bacteria starves itself of nucleotides. We see a highly targeted metabolic attack in treating malaria. The drug artemisinin circulates harmlessly in the blood until it encounters the malaria parasite. The parasite stores highly concentrated iron from digested red blood cells. When artemisinin hits that iron, it reacts violently, creating free radicals that shred the protozoa. Destroying a pathogen doesn't always require a direct assault on its walls. Cutting off a unique food supply provides a quiet, highly selective strike. But microbial biology adapts. Every time we deploy one of these strategies, pathogens leverage the resistome, a massive pool of genetic countermeasures used to fight back. Look at this text box detailing biochemical mechanisms of resistance. Bacteria produce enzymes like beta-lactamase, which inactivate penicillin before it reaches the cell wall. Pathogens also alter internal binding sites or construct molecular efflux pumps to spit antibiotics right back out. These genetic defenses have led to the rise of multi-drug resistant strains, like MRSA and the escape pathogens. Against these heavily armored microbes, our traditional magic bullets bounce right off. Selective toxicity is a fragile advantage. Every antibiotic we deploy acts as an evolutionary filter, killing the weak and inevitably breeding harder-to-kill survivors. To combat this severe resistance, modern medicine uses combination therapy, sometimes called sequential blockade. When treating complex diseases like HIV or tuberculosis, doctors administer a cocktail of multiple drugs at once. This forces the organism to attempt to survive multiple independent biological failures at the exact same time. The strategy relies on raw statistics. A pathogen might easily mutate to resist one specific mechanism, but the mathematical probability of it simultaneously mutating to survive 2 or 3 different mechanisms drops to near zero. This is a continuous race to identify and target new structural vulnerabilities before pathogens adapt to our existing arsenal. We are constantly searching for the next biological weak point in an environment that never stops changing.